All right, so let's continue this methods of harvesting energy lecture by getting into photosynthesis. So you are a heterotroph. You consume food to get your energy because you do not have the organelles in your cells to conduct photosynthesis. Heterotrophs consume other organisms that already have energy in their cells. All animals do this, all fungi do this. In contrast, an autotroph is an organism that is able to use some type of organelle, typically a chloroplast, but there are some bacteria that can do something called chemosynthesis instead. But there's some type of organelle that's used to produce energy-rich molecules using an external source of energy. And so what these guys do, autotrophs, is they start with an inorganic source of carbon like CO2 and utilize an external energy source to convert that CO2 into usable organic compounds. If you do this using the sun, you are what's called a photoautotroph. Photo means light. Photo equals light. So anytime you see the word photo, it means light. Photoautotrophs use the sun's energy, solar energy, as their source of energy to convert CO2 into organic compounds. Most plants are this. There are a few strange exceptions, but most plants are autotrophs. Algae, including single-celled green algae and large brown algae like kelp, which is actually not a plant. And there are some single-celled protists like euglena, which is this one, that can move, but they're also photosynthetic, which I think is pretty cool. So those are photoautotrophs. And if you're a photoautotroph, you're going to be green. Because chloroplasts have chlorophyll, which is the pigment that makes them green, and they cannot conduct photosynthesis without that chlorophyll pigment molecule. There's also a group of bacterial cells called cyanobacteria that conduct photosynthesis. So they are bacteria, they're prokaryotes, and yet they conduct photosynthesis. They actually do not have chloroplasts, to my understanding. They have a different method of doing this, but it's pretty cool. If you remember back from before, exergonic, endergonic. Endergonic reaction starts with reactants that have very little potential energy, ends up producing products after a chemical reaction that have a lot of potential energy. In the case of photosynthesis, we start with CO2. We end up with sugar. Energy is not created nor destroyed, so energy doesn't just show up in sugar, it has to come from something. And in this case, the external energy source is going to be the sun. Right? If you're a photoautotroph conducting photosynthesis, your energy source for this is going to be solar energy or the sun. Very good. So, chloroplasts. These are the organelles that, other than cyanobacteria, every single photoautotroph has. It's a specialized organelle that is able to harness the sun's energy, but it cannot conduct photosynthesis without the presence of a particular pigment called chlorophyll. This is the pigment that's responsible for the green color that we see in plants, and chlorophyll is the actual molecule that harnesses that incoming light energy. So if a part of a plant is not green, it can't do photosynthesis. Chlorophyll has to be there for a plant to conduct photosynthesis. So if part of a plant appears green, it almost certainly is going to have chloroplasts because it also has chlorophyll. Let's take a look at a leaf here. So when we look at leaves, we look at them like this. But let's imagine that we took that leaf and we put it on its side so that it would be extremely thin to look at. You're looking at this thing, flipping it like that. If you magnified it quite a bit, you would see this. The top layer and the bottom layer of the leaf are the top and bottom, or upper and lower, I should say, epidermises. These provide protection and help retain water for the plant. Then you have that middle layer right here. All of that is called the mesophyll, which means the middle leaf. The mesophyll is where the chloroplasts are concentrated. There are so many of them that it makes the whole leaf look green. The chloroplasts are concentrated in this middle region called the mesophyll. So that's the part of the leaf that conducts photosynthesis. Also notice there's a gap. Here's the bottom. This gap is called a stoma. It's a Greek word that means mouth. This is where you're going to have CO2 enter the plant. It's also where oxygen is going to leave. Okay? But it's called a stoma. Plural is stomata. This is like the plant's mouth. And on the side here, you've got these things called guard cells that regulate whether those stoma pores are open or closed, depending on perhaps the heat, trying to retain water, etc. If you look at the top of a leaf, and I'm not sure you can see it very well with the lighting as it is, here let's temporarily dim the lights. These are the stomata, and surrounding the stomata are the guard cells. I'm curious how dark this looks on the phone. Yeah, you can't read the screen very well like that. We need the light on. But those are the stomata, right? They allow CO2 to enter the leaf and oxygen to exit. And surrounding the actual pore itself are the guard cells that regulate the opening and closing of the stomata. So this is kind of the structure of a leaf, which is the primary photosynthetic part of a plant, right? Any part of a plant that's green is typically going to be doing photosynthesis. And so for a lot of larger plants with woody secondary growth, like trees and shrubs, etc., the leaves are the primary photosynthetic site. But some smaller plants that have green stems, right, those stems would also be carrying out photosynthesis. Let's look at a structure of a chloroplast. So we figured out that they're concentrated in the mesophyll section of leaves. Here's what one chloroplast looks like. You can see the picture up on the screen. Chloroplasts are surrounded, first of all, by a double membrane, and you can see that right here. Outer membrane, inner membrane. Just like with the mitochondria, except that there aren't any folds in the inner membrane of the chloroplast. The tannish, yellowish part, beige I guess maybe is the right term for it, this part here, this is called the stroma. Again, please note that R, it's different from the stoma. It's called the stroma. It's a fluid, right? It's a fluid. And within the stroma, you've got these structures called thylakoids. Here, I'm going to outline one of them. That's one thylakoid. It's almost like a thick, hollow, coin-like structure. The outer membrane of the thylakoid has all of the chlorophyll. That's why I colored it green. The inside of the thylakoid, this part right here, the white part, that's actually hollow. It's open. It's called the thylakoid space. So you've got the outer two membranes, the fluid stroma, the thylakoid membrane, that's the green part, and then the thylakoid space, which is the white part inside of the thylakoid membrane. So each of these little coins is called a thylakoid. Keep that in mind. Since the thylakoids have the green chlorophyll, that's where the actual light harvesting part of photosynthesis has to happen. Can't really see this picture very well, but it's just a cell with a bunch of chloroplasts inside. Multiple cells. So let's go ahead and answer this question then. Why do trees appear to be green to us? Why do trees appear green? If you said chlorophyll, you're right, but let's go into a little bit more depth on that. This is a sweet gum tree, by the way. Trees appear green because of the way that chlorophyll reacts to visible light from the sun. So, the sun emits electromagnetic radiation in the form of waves. And different types of radiation have different wavelengths. And let's draw something to help you visualize what wavelength refers to. Here is our sun, hypothetically. It's going to give off different forms of radiation with different wavelengths. Wavelength, I'm going to use red for it, is the distance from crest to crest of a wave. Notice how, as we go down this direction, I have increased the wavelengths. The shorter the wavelength, the higher the energy present in that radiation. There's less energy in waves with greater wavelengths. Only a small portion of electromagnetic radiation from the sun is actually visible to us, and that's visible light. Let's take a look at the whole electromagnetic spectrum. We'll come back to that. Here is the whole spectrum from the most energy over here to the least amount of energy over here. And these are the actual measurements of the wavelengths of the different types of radiation. We are interested in everything between these black lines because much of the other wavelengths that we have here are not relevant to this topic. But the things between those two lines are. Infrared radiation primarily speeds up the motion of molecules. We perceive this as heat. Right? Infrared radiation has wavelengths with less energy than visible light. So if we're using this picture as an analogy, these two right here, these two waves, we could put those as, hypothetically, infrared radiation. UV, ultraviolet radiation, has greater energy than visible light, and so we can put that up here as UV radiation with greater energy, shorter wavelengths. That leaves us with a small snippet of wavelengths in between those two called visible light. The wavelengths are longer than UV, so they're less damaging, but they're shorter than infrared. In fact, one of the reasons UV is so damaging is because of the energy in those waves. It can be so energetically powerful that it can actually damage your cells and cause mutations like melanoma, skin cancer. The visible light, this is the one plants can use for photosynthesis. It's the one that we can see. It's a small range of wavelengths between 380 and 750 nanometers. So that's very, very small. But notice in this picture then how different wavelengths correspond to different colors. So we've narrowed down with our picture here that we're looking at the visible light part of the spectrum, just a small snippet of wavelengths between UV and infrared. I do not have purple, so we're going to use blue, green, and red for my examples here. Blue light has a wavelength roughly around 450, looks like. So, we're going to make blue light look something like this. Green light has a wavelength around 520 or so, greater than blue. Notice how the crest to crest measurement is greater for green, because the wavelength is going to be longer. Red is up near 700. Crest to crest is a much longer measurement. And of course, yellow, orange, violet, all of those would also be up here as well. But you get hopefully the visual from that of how this works. Different colors correspond to different wavelengths. So what is chlorophyll? It's a pigment molecule. It's going to absorb certain wavelengths and reflect other ones. What we actually see is the color that is not absorbed. So going back, chlorophyll is going to absorb all of these colors just fine, and then won't absorb those very well. And it'll absorb these just fine as well. The weakest absorption is going to occur in this general area, which means most of that gets reflected. We see what gets reflected because chlorophyll poorly absorbs green light. Chlorophyll is a pigment molecule that actually harnesses light, and chlorophyll does not absorb wavelengths very well between 520 and 600. So, what do we see? We see what it does not absorb, which is green light. This is going to be where we do not perceive. Sorry. This is going to be what we do perceive. I got distracted by a car out there. This is going to be what we do perceive because it's what's not absorbed by the chlorophyll pigment. So that leads to another question. Trees change color in the fall. At least some of them do. Deciduous trees do. Those are the ones that shed their leaves on an annual basis. That's a maple leaf. It's yellow. Why is it yellow? Oh. So, in the fall, the temperatures and the day length are not really sufficient to keep doing photosynthesis as the plant had been doing all spring and summer long. So, there's no point in using energy to make chloroplast when you can just go dormant as a plant during the winter. So chloroplast production ceases, the chloroplasts are broken down, and for a short amount of time, the other pigments that have always been there are still visible. Here's a good way to look at that. Let's say that you are looking from above at this hypothetical forest. Pretend these are trees. Everything's going to look green when you look from above. But let's say that underneath the forest, we've got a bunch of smaller red trees. From above, it's still going to look green. But if you get rid of all of the green in the fall, you'll see that other pigment that has always been there. And depending on what other pigment is there, that determines what color the tree is going to look like. So once photosynthesis is no longer conducted in the fall, chloroplast and chlorophyll break down, which allows other pigments to become visible to our eye. Xanthophyll is one such pigment. Xanthophyll is a pigment that tends to reflect yellow wavelengths. This hickory tree shows that it has xanthophyll as a secondary pigment. Anthocyanins tend to poorly absorb the red wavelengths, and so some trees like this sumac tree reflect red wavelengths. Carotene and other carotenoid pigments tend to reflect orange light and red light, and so you get a lot of maple trees that have high concentrations of carotene as a secondary pigment. So you have all these secondary pigments. That takes energy by the plant for the plant to use to produce them. Why would you waste your energy on a secondary pigment? One hypothesis is something called photoprotection. Photoprotection basically states the following. Secondary pigments, that's these three. Secondary pigments absorb potentially damaging UV radiation. That is one of the dominant hypotheses as to why they're in there. They take potentially damaging UV radiation, and if they experience a mutation, that's not going to affect the plant's success in terms of its biological fitness in the same way that it would if chlorophyll experienced a mutation. So they think that these offer some type of protection from potentially damaging UV radiation. That's the current hypothesis. So let's look at photosynthesis stages. It tends to occur in two distinct phases. Not tends to, it does occur in two distinct phases. If you get nothing else out of how photosynthesis works, I want you to comprehend this picture. Okay? We can split this into the two stages like this. First half, second half. The first half is called the light reaction. Brilliant naming scheme because it requires sunlight directly. The light reaction needs a couple things to work. Number one, it needs sunlight. Secondly, it needs water. Thirdly, it needs chlorophyll. This half of photosynthesis is the one where the plant needs sunlight, water, and the green pigment chlorophyll to harness the sunlight. However, no actual sugars get made in this step. Instead, what gets produced are energy-rich intermediates, ATP, and an electron carrier called NADPH. It's very similar to NADH, which we talked about in cellular respiration. This one just has an extra phosphate group. So it's nicotinamide adenine dinucleotide phosphate. Point is, it does the same thing as the NADH. It carries electrons and serves as a mobile energy source. It moves the energy in those electrons. So the light reaction of photosynthesis, the first half, happens in the thylakoids of the chloroplast because it needs chlorophyll to work, harnesses sunlight, uses water, produces ATP and NADPH, which have a lot of energy, but they're not sugars, but they have a lot of energy. It also results in the waste product of oxygen being produced. The Calvin cycle, the second half of photosynthesis here, requires carbon dioxide and actually churns out a sugar in the process. But in order for this to happen, we have to have the energy-rich products from the light reaction to do it. So the sugar making itself is the second half of photosynthesis. It does not directly require sunlight at all. But in order to work, it needs the products made in the first half of photosynthesis, which is the light reaction, which does require sunlight. Light reaction, Calvin cycle. Two halves to one photosynthesis process. Let's go ahead and talk about the light reaction. This takes place in the thylakoids because the thylakoids have chlorophyll. The reaction requires the use of chlorophyll to absorb packets of light energy called photons. So we talked about light as a wave, right? Because it was we talked about wavelength and all that. But light also takes the form of particles called photons. Wave particle theory. It's physics. Don't ask me about it because I don't know. But it's a packet of light energy. All right, that's what a photon is. I think it's best to look at the entire picture before we talk about the actual stages. Here it is. Oh boy, this looks just like the cellular respiration stage 4 that we talked about. I'm going to go through this, and hopefully this will make sense to you. Number 1, light energy hits the plant. Let's talk about where we're actually looking at in the plant here. Here's a chloroplast, right? There's our double membrane. Here is a thylakoid. Remember, it's a stack of hollow coins built from thylakoid membrane surrounding open space. All of this fluid out here, this is the stroma. These are the thylakoids. Thylakoid space, green thylakoid membrane. This picture is showing you a snapshot of right here. Right? Up here, all of this is the stroma. This is the thylakoid membrane. And this is the thylakoid space. Perfect. So, what is going on? In the thylakoid membrane, we have these purple protein systems. They're called photosystems. And yes, it is just what it looks like. Photosystem 2 occurs before photosystem 1 chronologically. Awesome. So, photosystem 2 was actually discovered after photosystem 1, and they just kept the name. Photosystem 2 is the first one to receive light energy. This is where water comes in, another reason why you need to water your plants. H2O jettisons the oxygen atom, jettisons the protons from the hydrogens, which leaves us with 2 electrons. The electrons, one at a time, are going to enter this thing called the photosystem. Do you guys remember, and I hope you do, the energy staircase from the last chapter? I guess it's technically still this PowerPoint, but from the last chapter. Is that showing up on the camera? Yes, it is. Perfect. The energy staircase, if you remember, high energy, low energy. Water, the electrons found in water have no potential energy. You can drink all the water you want, you're not going to get any calories. What happens is that the water molecule donates the electrons to the photosystem devoid of energy. These green circles are chlorophyll molecules. They harness light energy and direct that light energy ultimately toward the center of the photosystem, where a special pair of chlorophyll A molecules are there with the electron that water has donated. Sunlight energizes the electron. So, sunlight hits the electron. It got to it because chlorophyll was able to harness it. And when the sunlight energizes the electron, it's like a springboard. The electron goes from the bottom of the energy staircase to the top. It has energy now. Basically, the sunlight took energy and put it into an electron from water. It's the long and short of it. So, now we have an energy-rich electron. That electron will go down the staircase. If you remember from the previous chapter, it's going to be passed between a bunch of electron carriers and release energy in the process. Just like before, that is going to be used to pump protons into the thylakoid space, and then the energy of the protons moving across that membrane into the stroma is going to be used to make ATP. It's the same mechanism as the electron transport chain, right? It's chemiosmosis all over again. ATP is the energy currency of the cell, which means that we've got a nice source of energy here that's going to go off to the second stage of photosynthesis, which is the Calvin cycle. But we're not done yet here. Not quite yet. By the time the electron releases all of its energy to be used to pump these protons against the gradient into this membrane space, by the time it's done with all that, the electron has no energy again. It's back to the bottom of the staircase. And so we need a second photosystem, which is actually called photosystem 1. We need another photosystem to harness light energy again and do the same thing as we did before. Take the electron, energize it, and send it back up the staircase again. However, this time, once the electron is at the top of the staircase, it never goes down the staircase again. Instead, it gets snatched up by NADP plus, which, once it snatches up the electron, becomes NADPH. That has a lot of energy now. The electrons are a mobile energy source. And so by the time this is all done, we've created NADPH and ATP, energy-rich molecules that are ready to go off to the Calvin cycle and actually make the sugars there. No sugars have been made yet. Oxygen given off by plants in part comes from the oxygen that is released from water that they break apart at the beginning of this whole light reaction. Let's see what we're doing for time-wise on this lecture thing. 32 minutes. Okay, let's stop it there. Actually, we'll stop it there and start up the new lecture. We are back. Part 2 of photosynthesis. Let me just tell you guys, I have not eaten. It is 1 o'clock as of this recording. I have not eaten since this morning. I'm absolutely starving. But what's new, right? I figured I should mention it to make it feel like you guys are truly back in this classroom, because I feel like I talked about food a lot in class. All right, the next couple slides here summarize everything that I said. I'm not going to read through them, but if you want, I highly recommend to look at the PowerPoints and kind of work your way through all of this if you're somebody who would learn it by reading as well. So we got ATP and NADPH, energy-rich molecules ready to go to the Calvin cycle. This occurs in the stroma of the chloroplast. So if you remember the chloroplast structure here, right? We had the thylakoids, but then the stroma was the fluid that surrounded them. So we made ATP and NADPH using the light reactions in the thylakoids, and then those get sent out to the stroma where the Calvin cycle is going to happen. In order for the Calvin cycle to happen, we need ATP and NADPH, and we also need CO2. We cannot produce sugars without an inorganic source of carbon first. So CO2 is necessary for the Calvin cycle to happen. What happens in the Calvin cycle? Carbon dioxide, CO2, is converted to something called G3P, which is a sugar precursor. We're not going to talk about the technical way in which G3P becomes glucose, but G3P is effectively a prelude to sugar. I think looking at the whole picture is going to help here as well. So here we go. It's a cycle, first of all, which means that something always has to be regenerated and continuously move around the cycle. The name of the molecule that's regenerated is called ribulose biphosphate, typically shortened to RUBP. There are 3 molecules of this that are coming back around the cycle every time it churns. An enzyme called Rubisco is necessary to facilitate the incoming carbon dioxide into the cycle. Think of it almost like merging onto a freeway, except that you want to hit the car that's coming back around, or coming on the freeway. Yeah, that was a terrible analogy, but never mind, let's continue on. 3 carbon dioxide enter the cycle. 3 molecules of ribulose biphosphate are coming around the cycle. Rubisco is going to join them. It's the most abundant protein on the planet. Think of how many plants there are. Makes sense. Rubisco configures the incoming CO2 with the ribulose biphosphate and ends up producing 6 phosphoglyceric acid molecules, or 3-PGA. So we have 6 molecules of this PGA intermediate that occur once the CO2 coming in is joined with the RUBP that is being regenerated, that is going back around the cycle here. That's the first step of the Calvin cycle. It's called carbon fixation. Fixing something means that you're putting it into this cycle. The 3 molecules of PGA, they're the ones who are going to be energized using our products from the light reaction. So if you remember, in the light reaction, we produced ATP and then NADPH carried some electrons. Those both have a lot of energy, and they release it and change our PGA molecules into 6 molecules of G3P. The technical name for this step is called reduction. Might be a strange name if you're not familiar with the term, but the word reduction actually means that you are gaining electrons. So NADPH is dropping off its energy-rich electrons, and thus the PGA molecule gains them. And so reduction involves the gain of electrons. And so by the time all of this energy is released into our cycle, we've got 6 molecules of glyceraldehyde 3-phosphate, or G3P. Step 3 of the Calvin cycle involves one of those 6 leaving the cycle. One of the 6 leaves the cycle and is used to make glucose, fructose, proteins, etc. It's used to make organic compounds for the plant. The other 5 go back into the cycle, and with the assistance of some ATP are reconfigured into ribulose biphosphate again. That's step 4. Step 3, by the way, was when that one G3P left the cycle. Step 4 is regenerating RUBP so the whole thing can happen again. 3 molecules of carbon dioxide are required for this cycle to churn once, and it's churning thousands upon thousands of times as the plant is incorporating CO2 to produce sugar molecules. None of this works without products directly from the light reaction. However, This can actually take place in the absence of sunlight, and that's just fine. It works just fine that way. So this is Calvin cycle, which is stage 2 of photosynthesis. The next several slides go over the stages. Highly recommend that you look those over and read the text. It should say exactly what I told you. So as we mentioned before, plants take in carbon dioxide and get rid of oxygen through pores in their epidermis called stomata. So here's a stoma, which is the singular form of stomata, and it's a nice pore for oxygen to leave and CO2 to come in. But there's another issue with that. When it gets really hot out, you're going to lose a lot of water through transpiration, which is the plant equivalent of evaporation, basically. And so a lot of water potentially may leave out of the stoma. That's a problem. When it's hot out, you got to conserve water. In hot weather, certain plants will close the stoma to minimize water loss. Sounds like a good plan. So the guard cells here are going to close, which means water stays in. Unfortunately, it also means carbon dioxide can't come in, and oxygen can't leave. So you end up with too much oxygen inside the plant and not enough CO2. Now, if we look back at this slide, right here, you see Rubisco is supposed to fix the incoming CO2 with the RUBP, but Rubisco is not very good at differentiating between oxygen and CO2. So if there's too much oxygen present in the plant, Rubisco will tend to use oxygen instead of CO2, which doesn't actually make sugar. It wastes energy, and over time the plant is effectively going to die from that because it's wasting energy and not actually producing sugars like it should. So in most plants, they build up too much oxygen when they close their stomata. The plants begin using O2 instead of CO2, which makes them waste energy and not actually produce any sugars. It's called photorespiration, and it is very potentially damaging to the plant. It can result in death for the plant. So C3 plants, most plants, do not do well in hot, dry environments because of this, among other things. But this is a big problem. Most of our plants here are C3. However, there are plants that have ways to deal with it. One way is to be a C4 plant. C4 plants are able to keep their stomata barely open and still take in plenty of CO2 that way. Think of it this way. We've got a stoma here with barely any space between, right? And then we've got one here that has to leave quite a bit of space between in order to function. If you can have barely any space open, you're not going to lose a lot of water compared to this wide open stoma here. So C4 plants are able to keep their stomata more tightly closed. Corn and many species of grasses fall into this category. Another strategy is to be a CAM plant, a CAM plant. It stands for crassulacean acid metabolism. What these guys do is at night, the CAM plant keeps the stomata open, takes in a bunch of CO2, and doesn't lose much water because the sun is not out and it's a lot cooler at night. During the daytime, these plants will close their stomata but they have banked enough CO2 that they can do the light reaction and the Calvin cycle during the day when there's sunlight available and they don't lose any water. There's no excess oxygen buildup because at night, when the pores open, the oxygen can leave. So CAM plants only open their stomata at night, taking in CO2 at night, banking it, and then using it during the day. It's a pretty good strategy. These plants are pretty darn successful in desert environments. Cacti, succulents, fall into this category. Roughly half of the sugars produced by photosynthesis are going to be broken down and converted into ATP so that the plant can conduct cellular respiration and do physical work. If the plant has excess sugar, it might store it in its tubers, in its root system, like carrots and beets. It might put it in fruit. It might create nectar with it. Also keep in mind that plants are not just carbs. I don't know if you guys have eaten peanuts anytime recently, but they're protein and fat. Both of those are organic compounds that the plant can produce using photosynthetic products as a starting point. So the plant produces sugars, and then it has mechanisms to convert those sugars into other organic macromolecules, like fats and lipids, or like lipids and proteins.